कॉफ़ी दे दो। वाला नहीं चाहिए था पॉसिबल मट्ठी या कुछ ले दो अंकल दुकान पर फैन मट्ठी So, good morning, students. Yesterday, we were done with the DNA structure, and we have discovered that the basics of DNA, the how the nucleotides, nucleic acids, they all make the DNA structure. Yeah. The diameter around 200 nanometer, major groove, the length of major groove around 2.2, minor groove 1.2, length of one turn 3.4. This includes around 10 nucleotides. Distance between two nucleotides is known as rise. Uh, so we have discussed many number of things yesterday. So based on that, we are having three types of DNA, that is A, B, and Z. The A DNA, it's a right-handed double helix, and B DNA is also a right-handed double helix. These things are common in them. The major difference is that it has 11 nucleotide base pairs, your A DNA, whereas the B DNA is around 10 base pairs per turn. And the diameter is quite bigger than the B DNA, that is 2.3 nanometer. Here, the diameter is around 2.0 nanometer. And this is widely accepted DNA in the all living system as compared to the ADNA. So this is much broader, we can say, from the length perspective, like 2.3, and this is not so much. And this is even thinner, that 1.8 nanometer, but longer, 12 base per turns. And this is called zDNA because instead of having this helical structure, this structure shows a Z-tape transformation, like this. And this is an exceptional, that is left-handed double helix, and it's also very less common present in the living system, not so common one, the zDNA. So these are your ADNA and BDNA, both are right-handed helix, and this one is only the left-handed helix. So in this way we can see, but this one moving upwards. What else we can see? That it's bit thinner, the diameter. This is middle, and this is broader. This is quite broader. And here we have in the one turn 11 nucleotides. Here we have 10 nucleotides per base pair turn, and here we have 12. So this is the longest. This is more stretchable. This is in between. This is in between somewhere, and this is the smallest, BDNA. Yeah. So that's the major differences that we have just discovered. Also, there's a difference between the major groove and minor groove. We will discuss one by one each of them. And before we go ahead, just want to discuss that in the bDNA we can see that minor groove is smaller, yeah, as in the rituals mostly, and the major groove has quite deep and thick. But in the aDNA, the minor groove become more deeper and the major groove is opposite. So it's opposite of the bDNA. Now also there is one term I have discussed, the rise, that is the distance between your two nucleotides, and there is another term called tilt. Tilt is the angle between two nucleotides. Tilt is the angle between the two nucleotides, and it's different from each um, in each DNA. Yeah. So let's take everything one by one. First is your overall proportion. That is alternate forms of DNA, that is aDNA, bDNA and zDNA. So the aDNA, it is more short and broader, and this is longer and thinner. This is elongated and slim. Now why they are short and broad? Because the rise per base pair, like the distance between two nucleotides, is 2.3 angstrom. Yeah, and here is 3.32 plus minus 0.19, and here is 3.8. So that's why it's the most elongated. This is okay, longer, middle longer, and this is the shortest. And the diameter, if we see, this is the most highest diameter, that's why it's broad. This is short, this is broad, this is longer, this is a bit thinner, this is elongated, and this is even slimmer. Based on these two values, we having of the overall proportion of our three different types of DNA. Now let's take to the helix rotation. This was first, this is second, this is right-handed, this is right-handed, this is left-handed. Right-handed, right-handed and left-handed. Base pairs per helix repeat. So number of base pairs in the helix repeats are 1, 1, 2. And as we have discussed just now, in the aDNA it's 11 and 10 and 12 number of nucleotide per turn. Also tilt, perpendicular tilt, it's 19 degree plus 19 degrees Celsius here, and here is minus 1.2 degrees Celsius or degree, and here is minus 9 degree. So that's the tilt differences between three of them. Major thing that we have to think here is the major groove and minor groove. The major groove is extremely narrow here, but very deep. So this is opposite of the bDNA. Here is wide with intermediate depth. And on the other hand, the zDNA is flattened out on the helix surface. Whereas the minor groove is very broad but shallow. This is opposite of the bDNA, whereas in this case, the minor groove is narrow with intermediate depth. And here is extremely narrow but very deep. So somehow these two are like same in this case, but this aDNA, the major groove and minor groove, shows a complete difference as compared to your B and Z DNA. Yeah. So this table, whenever there is any subjective exam or objective exam, or you have net exam or gate exam is coming up for you, so this table really explain each part of your aDNA and bDNA and zDNA. So make sure you go through it, understand each terms what they are, with the help of diagrams and what their meaning. Yeah. Here we can see this is Z-type structure. This is very nicely. This is even some not so good structure. We can say the aDNA. So that's the major difference between your aDNA and bDNA and zDNA. Okay. So now we will go to the next part. That is in another in eukaryotes and prokaryotes, how they are. In eukaryotes, they are mostly linear and they have ends are open. Where in the prokaryotes, it's circular and the ends are not open. So in the prokaryote, this is circular DNA. In the eukaryotes, we see this, which has free ends. So that's the major difference between eukaryote and prokaryotes of your DNA. So answer these 10 questions, students, into the chat box. DNA stands for, this is a revision of what we have done. What is the shape of DNA? Who established the structure of DNA? Adenine always pairs with. The sites of DNA ladders are deoxyribose and. Okay, first answer first five, then we go for next five. Answer these first five questions in the chat box, and I will check them how they are. Then we go. Then we proceed. Hurry up, student. Answer them quickly. Okay. Deoxyribonucleic acids. It's a double stranded helical. Yeah, double stranded DNA. Double helical. Yeah. Watson and Crick, thymine, phosphate group. Good. Very nice, Sharvari Pujure. Very nice. Good, good. It's helical, but not B form. Sumesh. Thymine, phosphate. Okay, now answer the next five. Guanine always pairs with what is the complementary sequence towards this sequence? The two strands of DNA are held together by which bonds? And DNA is composed of repeating subunits called, and which are the four basis pairs which makes the DNA ladder? Very simple, very fast, just answer them. Then we go ahead with the basics of a little bit about RNA, because we have discussed DNA in detailed manner now. Now it's time to discuss about. Answer them quickly, students. Think about this. I'm asking two DNA are held together. Answer fast, fast, hurry up. Okay, good. So, did I share this video already with you? So, you have gained understand from this lecture the structure and function of DNA as a genetic material. So, based on that, we were able to understand replication, transcription, and translation in our upcoming lectures. So, thank you very much. Yeah, structure of DNA has been shared already, and we have discussed about the cell size. now I wanted to take you to the next part of RNA world. So we'll not discuss, because we know that the basic components so far for the in the field of molecular biology or biochemistry, it's the DNA, RNA, and protein. So they play a central dogma of life, where DNA is producing your RNA and RNA is producing your protein. Now, so gene, which is acting as a unit of heredity here. So DNA segments that carries genetic informations are called genes, right? And it is normally a stretch of DNA that codes for a type of proteins or for RNA chain that has function in the organisms. And genes which hold the information to build and maintain organism cells and pass genetic traits to offsprings. So here we have our nucleic acid, our DNA, which is coiled around the histones, and these histones get coiled around and make a chromosome, sister chromatids of your chromosome. And here within, and these chromosomes are present in our nucleus, around 23 pairs. And within these chromosomes, our gene 1, gene 2 are present, which go to the further offsprings. So this DNA we have discussed in detail now. I will not go through them. If you want to have these notes, you can keep them. So my part is here now with the RNA. So what is a ribonucleic acid? So this is actually a very important part also after the DNA. If RNA is not being produced in our body, we will not survive. So that's why it's very, very important. Very, very important. So it's one of the biological important type of molecule that again consists of long nucleotide units, nitrogenous base pairs, your ribose sugar, and the phosphate group. But in this case, they will be not double stranded mostly. It will be sometimes single stranded, and then it's again become a double stranded. So in this way, again single stranded, again double stranded. In this way, our RNA molecules looks like. And in viruses, somehow the exceptional, they are also double stranded. They are found out as a dsRNA. So there are three different types of RNA according to their functions. First is messenger RNA, then the ribosomal RNA, and the transfer RNA. This is abbreviated as messenger RNA, this is as a ribosomal RNA, and this is as a transfer RNA. And this functions, that is codes for protein, and this is a translation, and this is also for the translations. And they are mostly present in all organisms. Okay. And the case of post-transcriptional transcriptional modification, small nuclear RNA, snRNA, it's deal with the splicing and other functions, eukaryotes and archaea, and then the yRNA and telomerase RNA. So they are used for RNA processing and telomere synthesis, and they are used for animals and most eukaryotic organisms. And regulatory RNAs, so they are antisense, that is transcriptional attenuations, mRNA degradations, and translation block. So that's the complete chart about different types of RNA here in summary. So let's take one by one. Messenger RNA. So that's basically carries the information about a protein sequence to the ribosomes and where the protein synthesis is happening. So it is coated with 3 nucleotides and correspond to 1 amino acids. So we will discuss them in a very good presentation just now. So in eukaryotic cells, one precursor's mRNA has been transcribed from DNA. It is processed to mature RNA, and this removes the introns, non-coding sections of the pre-mRNA. So within this mRNA, they have coding sections and non-coding sections. Yeah. So messenger RNA role is actually to remove the non-coding sections. So these non-coding will be removed, that is your introns, and mainly your exons in the, it's called splicing. During the uh, splicing step, only the exons, which are coding regions, will go further. So here we can see the DNA is unwinded, and it will make one is non-coding strand and other strand will be the coding strand. And our mRNA will start to produce in corresponding to all nucleotides. Like for C is G, for A it will be U, A for U, it will be not T here. Yeah, don't think that T will be producing here against of that. That will be producing during the DNA replication. But during the transcription, A will produce U and T will correspond to A. Remember this. So the mRNA is then exported from the nucleus to cytoplasm, where it is bound to ribosomes and translated into the corresponding protein with the help of tRNA. Before I go further in details, I want to take a couple of animations now, because I always tend to have the lectures. We have at once some lectures, then some videos, some animation, some virtual labs. So we will have a combination of trio together. It's not just these I keep speaking about the lectures because this sometimes to make it's very hard to absorb for the students also then. So in this line, it's just saying, summarizing the whole role of your transcription and translation, that mRNA after its work is done goes out of the nucleus, went to the cytoplasm, where it's bind to the ribosomes and then translated to the corresponding proteins with the help of your transfer RNA. Yeah. So I will stop here my presentation from here onwards, and I will take you to the next part. First of all, let's have an animation about DNA and genes. What are DNA and genes?
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
So that's why we are different from our sisters, brothers, friends, mother, father, just because of our tiny differences between your A, T, C, G. So it's mainly the 99% of every human being. The genetic material is same. It's just 0.1% or 1% is the reason that things are different. Make us different. So I will keep sharing these links once we are done with them. So I want to make you to the next video. It's about same gene and different organism. So it's stating that how should I say in the words? Like there is a jellyfish, yeah. It has a genes which glows in the dark, fluorescent gene. And what if we put that gene into the cells in order to see the organelles functions? Or we put them into the fish and it will start glowing in the dark. Or there's spiders are producing silk webs, and we took that gene that is producing that silk web and put into the yeast. And from the yeast, we can have a huge production of these silk webs artificially then. So it will be the same gene in different organism, but we are getting a different function out of it. Same function though, but from different organism. So that's where your biotechnology, your molecular biology, your biochemistry, all together playing a role and making it a sense. So I will start with that now.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
So I hope this makes sense. Same gene, different organisms. Let's discuss what our proteins are then.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
Mhm. Something about little bit about proteins. So after watching all these beautiful animations, I want to take you to now the statement that I was discussing before. Transcription and translation that was done by the mRNA. So I'm going to take my next 10 minutes to watch this now. So sit back and watch them with interest.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
Let's take a gene. What gene? Maybe this one.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So C will produce which one, students? Tell me. C will produce which one? Tell me, all students, tell me. It will be G. Then for T, it will be A or U. For T, it will be A or U. Is it A or U? I want to check your IQ on this because we are working on mRNA, not DNA replication. Students, hurry up. It will be A. A. For C, it will be G. For T is A. And for A, it will be, students. For A, it will be U. Yes. Like it goes on.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So that was about translation. So let's check. AGC. It will be AGC. Tell me, students, what it will be. AGC. Now we are working on the translation. So these are anticodons that are present on the tRNA, and corresponding to them is yours, what is it? Is your amino acids, and they will make a polypeptide chain of amino acids. With the help of, this is your ribosome, small ribosome and large ribosome unit, and in between is your mRNA that is came out of your nucleus into the cytoplasm. So for AGC, it will be serine, threonine, aspartic acids, or arginine. What do you say? Is it serine? Yes, it is serine. UUC, phenylalanine, serine, threonine.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
Okay. So that was all about today. Translation, transcription, protein, DNA, RNA in details. Any questions so far, student? Okay. This is the structure of tRNA, which is a small RNA chain of about 80 nucleotides that transfers a specific amino acid to a growing polypeptide chain at the ribosomal site of protein synthesis. So it has anticodons on top here, and this is the codon site of your mRNA, 5' to 3' end, right? So it has sites for amino acid atman, anticodons, region for codon recognitions, all are being taken care. And there's a ribosomal RNA that is just 70s unit and 80s unit. And this is a major difference between RNA and DNA. That is, it has deoxyribose sugar, it has ribose sugar, base uracil is uracil here, and here's thymine. At the second position, the hydroxyl group is present, and this is absence. And it's usually single stranded, and this is double stranded. Proteins, also known as polypeptide chains, we have discussed. It has around 20 amino acids which make a sequence of a genetic code. So this is the sequence of like lysine, valine, phenylaline, all these polypeptide chain, they come together, and these are the two cysteines that are making a disulfide bridges bonds very nicely, and they are making this quaternary structure of protein, and together they are making into the red. You can get this in the red kidney beans, your peanuts, pulses, fish, chickpea, eggs, red meat, chicken breast. So all these are the sources of your protein. And these are the DNA, RNA, and protein. As we have discussed, they are playing the central role of your biochemistry or molecular biology. That is, DNA could self-replicate. Then after the transcription, it will make RNA, and then after the translation, it will make a protein. Yeah. But don't never ever that in the central dogma, this will never happen that from your DNA protein could produce directly. This will never happen. This could happen that from RNA, a complementary DNA become produced with the help of reverse transcriptase enzyme. With the help of reverse transcriptase enzyme, I can't write this. Yeah, and also from protein, your RNA cannot be produced. So this is also no. So only case that is possible is that RNA could produce DNA, and DNA could also self-replicate. So this is the basics of your central dogma of the life. About the DNA replication, we have not much time now, I guess. We have 5 minutes left. So I will discuss this tomorrow about DNA replication and a little bit about transcription and translation that we have just discussed in the video, but it's also good to discuss things virtually from the subjective point of perspective, because we can express the videos nicely, but it's also good to know how to express them in a subjective manner. So we will do that tomorrow morning. So for today we are done of many topics. We have discussed various types of DNA since morning, and then we have discussed about various types of RNA. Then we have started to discuss about various animations related to that, and then we have been to the very core understanding of our transcription and translation with that animation. And recently we have just discussed about your central dogma of life. Yeah. So I hope please go through them, all of these lectures, read them. So I will share this lecture also with you, this introductory, so that you can have a look over it. Cool. The rest we do it tomorrow then. Okay, class, then thank you very much. See you tomorrow at same time, same place, to continue further learning. If you have any questions further, please write me on the chat box and on the WhatsApp. I will try to answer my best. Thank you. Bye bye. Wish you a nice day ahead.